With our final Lecture 7 presentation, we conclude with final lessons from apes. There are other insights from chimpanzee stone tool use. In West Africa, researchers have documented the formation of chimp nut processing sites. The results provide a blueprint for what this would look like at an archaeological site. This has led to reconsideration of some prehistoric data. Repeated use of a preparation site produces nutshell trash and 500 stone pieces, hammer stones, and accidental flakes. The chimpanzee sites and stone tools are very similar to artifacts from early human archaeological sites. On the left, we've got the stone tools from the Thai forest chimpanzees, and on the right, we have the stone tools from ancient humans from Swartkrans in South Africa. They look very similar to each other. Regional variation in chimpanzee tool use has raised an interesting question. Are we observing the origins of cultural behaviors, sometimes referred to as the origins of protoculture? Chimpanzee regional variation in tool use may be due to protocultural differences. However, the habitats vary quite a bit in ecology, and the differences we see in tool use may simply be environmental. Over in the West, where we have the Western common chimpanzees, we have typically wetter forests, more tropical forest. In the central area, we've got a mix of tropical and dry forests, and over in the east, we have very dry forests, woodland savanna type. And then pygmy chimpanzees, again, have a mix from rainforest to very dry woodland forest. Termite wanding occurs in chimpanzee populations in dry savanna and woodland forests across western and eastern Africa. Nut cracking with hammerstones is restricted to common chimp populations in the damp rainforests of western Africa. The other sites do not have the same types of vegetation and, importantly, nuts. Chimp regional variation extends to hunting behaviors, but again, there is ecological variation. Now, before I go further with this, you need to understand that many primates engage in hunting behaviors. The best, of course, are the tarsiers, but mouse lemurs, galagos, and lorises are also excellent hunters. Animal proteins comprise a significant proportion of their diets, and in the case of the tarsiers, 100% of the diet. Capuchin monkeys of the New World and marmosets and tamarins, also New World monkeys, also utilize high amounts of animal proteins in their diets. Old World monkey baboons hunt deer and flamingo. But remember that these Old World monkeys must make use of every calorie available in their dry habitats. They are obligate omnivores. They would prefer to eat more fruit, but it's not available. Hunting is different with chimpanzees. Animal protein comprises less than 10% of chimp diet, and only 1 to 3% of animal protein comes from the meat of large-sized mammals. For chimpanzees, hunting is less about diet than it is about the endeavor. They like to hunt. Hunting has been recorded at nearly every chimp site, and the favorite prey animal is the red colobus monkey. These are very aggressive monkeys, probably because they've been hunted so heavily by chimpanzees, and they are so aggressive that they can chase humans away from their study sites. Thai forest chimpanzees in Western Africa engage in cooperative hunting. It's actually kind of scary. What happens is chimp or two will climb up into the trees and follow a group of red colobus monkeys. They'll isolate one individual. The red colobus monkey knows the danger, and eventually it runs out of tree room and leaps down onto the forest floor where the rest of the chimps are waiting, and they'll grab it up, tear it apart, and share the food. Gombe chimpanzees, in contrast, in eastern Africa, engage in solitary, every chimp for himself hunting. They see a red colobus monkey and they go after it individually and hunt for it and catch it, eat it. Bottom line, there is regional variation in chimp hunting behaviors. In the West, cooperative hunting. In the East, solitary hunting. Are these differences evidence of protocultural variation? Again, we are confounded by differences in habitat. The cooperative hunters to the west live in dense rainforest. Individual hunters to the east live in open woodland habitat. Chimps may be compelled to follow different hunting strategies based on ability to see prey. The great apes have provided much insight into hominoid cognitive development. Some of the most interesting data come from ape language studies. The earliest of these captive ape studies naively began with attempts to teach chimps to vocalize, to speak just like humans. It wasn't long before researchers realized that chimpanzees simply do not have the vocal anatomy to speak as we do. The chimpanzee vocal box is located higher in the throat than is the human vocal box. So if you take a look at the human, the reason we can reach low notes is because the vocal box is located low in the throat. For the chimpanzee, it's very high in the throat. And so they can't reach those low notes that we can. And this means that they don't have the pitch, the change in tone that we have. And importantly, chimpanzee brains lack development of the Broca's area of the brain that controls human speech production. Broca's area is located in the motor cortex of the frontal lobe. So if we're at the front of the skull here, back of the skull here, over here is the ear region. And right at the motor cortex of the frontal lobe, right through this area here, is the swelling associated with Broca's area in us. It is on a closed feedback system with Wernicke's area, which is located near the sound receptors. When a loud noise goes off next to your ear, your face will automatically respond to that. This is the response that allows articulation of the mouth muscles that provide speech. So all around your mouth you have these small buccinator muscles and a whole bunch of other muscles, as well as control of the tongue. And your ability to make consonants is controlled by these facial muscles. The chimpanzees lack these motor pathways, and so their faces are not involved in speech in the same way that they are in a human. Without that control and response to sound, they are less likely to make sound as we do using facial communication, etc. Human speech also involves the version of the FOXP2 gene that codes for amino acids, genes that allow speech. The chimpanzee version of this gene does not target the same amino acids, the same genes. It codes for other outcomes, and we don't fully understand what those outcomes are. Researchers began to teach apes American Sign Language and now use symbol boards, which allowed the apes to invent their own words. Apes can recognize and utilize numerous nouns, places, people, things, and verbs. They absolutely love action terms, but they cannot acquire the rules of grammar needed for constructing a complete sentence. There are no articles, no pronouns, no conjunctions. Ape linguistic skills are at about the same level as a preschool child's. So if you go to the grocery store and you've got a 3-year-old with you, and the 3-year-old wants a candy bar, it may not say, "May I please please have a candy bar. It's likely to reach out and point to the item and say candy, want candy, something like that. And that's about the level that the chimpanzees and orangutans and gorillas get to. When we get to early fossil humans, we will see that they had ape-sized brains equipped with similar capabilities. So you've got the chimpanzee, it's at the correct size in comparison to these early humans. We've got the Australopithecus species at the top. So Australopithecus afarensis is the oldest, followed by Australopithecus africanus. And below we have the Paranthropines, or robust Australopithecines, Paranthropus ethiopicus, Paranthropus boisei, and Paranthropus robustus. All had ape-sized brains. Nobody had a brain larger than 550 cubic centimeters in volume. We will also see that the adaptation that initially separates the earliest humans from apes was bipedalism, not increased brain size. So if we take a look at Lucy over here, what do we see? We see a bowl-shaped pelvis, a medial angle to the femur, and a tibial plateau. When we take a look going past the Pliocene into a little more modern time, the Homo erectus has all of the features associated with bipedalism. So bowl-shaped pelvis, femur with a medial angle, platform-like or horizontally placed platform on the tibia. So the first thing that marks humans as humans in the fossil record is bipedalism. The brains, on the other hand, the skull, in many respects, are very much like those of a chimpanzee. Now you are ready for Lecture 8, Primate Evolution.